Hi friends, today we have the glomerular structure of the renal corpuscle. Previous lecture we already discussed about these things, but that was labeled. Now we have without labeling and further highly magnified image of the renal corpuscle. So let's go over to find the type of cells and the space present here and other cells here. Okay, so let's begin. So here we have further first of all we have the renal corpuscle. This is what the renal corpuscle, having the Bowman's capsule and the glomerulus. So here let's begin to find that. First of all, the distinguish, these are the red without any nucleus. Okay, so it is very easy to find the red blood cells because they don't have nucleus. So these are the red blood cells. These are all the red blood cells. So we can see, you can say that these are the red blood cells, RBCs. And we know that RBCs are present in the glomerulus, glomerular capillaries. So here we have the capillary, one capillary, second capillary. Okay, so another capillary here. So these are the capillary network having the nucleus. This is the nucleus of the capillary, nucleus and nucleus. So here we have which one? That is the glomerular capillary, and these are the endothelial cells. Endothelial cells, they have in the nucleus, endothelial cell or endothelium. Okay, and the next structure here we can see the space. It's a very fine space, and this space is surrounded by two layers of cells, visceral and parietal. These are the parietal cells. Okay, these are parietal cells. So here we have parietal cells, that is a simple squamous epithelium of the Bowman's space of the Bowman's. Okay, and here the Bowman's space, Bowman's capsule or Bowman's space. Okay, and in the next cell here, these are the mesangial cells. Okay, you can see these are the mesangial cells, act as a macrophage sometimes. Mesangial cells. In the next group of cell here, the podocytes. You can find here the podocyte line, the visceral layer of Bowman space. Here the cell we have, and here we have the podocyte. Okay, so these are the podocytes cell. And next cell here we can see the membrane. Okay, in case of this we can see here is the membrane, okay, and here also the membrane, here is the membrane. So these membrane act as a barrier for the filtration. If we really want to discuss, we can see the, okay, so this is what the endothelial cell. There is a fenestrated capillary network, fenestrated capillaries lining the endothelium. They have fenestrations, pore, okay, which provide a significant filtration. And below this we have the glomerular basement membrane, have the lamina rara and lamina densa, connective collagen fiber. This is made up of, and then we have the special podocytes. Okay, podocyte cells having the filtration slit and negatively charged, and there is the parietal layer. This parietal layer is squamous cell, simple squamous. So here we have space, that is the Bowman space. Here the parietal squamous cells, that here we can see the parietal cells. Okay, here these are the parietal, and podocytes, here we have the podocytes and their RBCs. Okay, this is the RBCs, and endothelial cell, we can see the nucleus, here the nucleus, and here the lumen. The cross section image like this. Okay, and if we know the structure, then there is a simple another point, that is the space. This is the filtration slit. The gap between the food process is called filtration slit, and the size of this is 20 to 30 nanometer, so it is very small. It will not allow all the molecules which are bigger than 30 nanometer or bigger size than the filtration slit. And fenestrated capillaries, they have fenestrations of about 70 to 100 nanometer, so it acts as a size barrier, and this one mostly acts as a charge barrier. So thank you for listening.